Oh, hi, everybody. Good to see you all again. Yeah, it's interesting because we've heard so many great comments about the whole lecture series. You know, this whole idea of the kitchen table talks for the whole community. We're getting a lot of great feedback from people around the world about how they really enjoy these lectures, find them fun and educational. And so we're really glad to hear that. That's why we're doing this. We're doing this because we think that the whole condensate community can benefit. We're all wrestling with the same scientific challenges. And so it's good to be thinking about these things together with everyone. So today we have Shahar Sukenik. He's from University of California at Merced. I'm really excited about having Shahar here. Got his PhD at the Hebrew University of Jerusalem and then did a postdoc at Urbana-Champaign, really focusing on microscopy, really thinking carefully about the interactions of proteins in cells, in live cells. And obviously over the years has gotten attracted by the condensate bug like all the rest of us. And 3 years ago, took his faculty position at Merced, where he is now an assistant professor. And really interesting work because it highlights the interactions between proteins and environment and their environments, both in vitro and in vivo, in a cellular context. And of course, you know, Shahar does this with an amazing arrangement, a range of technologies, live cell imaging and spectroscopy, computational modeling, other things. And so really applying a multidisciplinary approach. And that's a common theme, isn't it? A lot of the condensate lectures we've heard really rely on such an interesting blend of technology to tackle these challenging problems. And so in particular, he's been focused on, you know, homeostasis. And we all know the critical nature of that and how the metabolic rewiring affects proteins in a variety of ways and how relevant that is for cancer and viral infection and lots of other pathology. And of course, that leads to IDRs, which are such an essential part of this whole process. And so naturally, a lot of the work that Shahar does is related to understanding the way the IDRs and the other aspects of proteins lead them to form these very specific and functional and stable condensates. And so very exciting to have you here today. And your title this morning is Turning Protein Disorder into Function. Is the solution the solution? Floor is yours.
Thanks so much, Mark. So first, I'd just like to start by thanking Jill for this invite and Mark for that fantastic intro, which basically kind of turns my intro obsolete, but I'm going to try doing it anyway. So it's really great to be here this morning. And so I think we'll just jump right into it. And the place I'd like to start with is a slight apology in that while there will be some discussion of condensates, most of this talk we're going to kind of zoom in and really focus not on the collective behavior of proteins, but rather on the behavior of the monomer. And kind of like Mark said before, to understand the collective behavior of these molecules, it's really important to first form a good understanding of how they behave when they're on their own. So that's kind of our entry point. And so the first thing I want us to start thinking about is the structure of proteins. And so usually when we go to a textbook, and now it seems like, right, this animation is working, because it didn't before. Right, so when we look at a textbook and we think about kind of these traditional well-folded proteins, they all have a very specific shape. And these shapes kind of inform about, or structures inform on the function of these molecules. And an absolutely beautiful example is this pilus assembly mechanism that's been resolved using cryo-electron tomography by the Jensen lab way back in 2016. But you can really see how this is a molecular machine, right? You see stuff that looks like cogs and parts, and they're moving in synchrony, and they're acting together to form this pilus, which is kind of like this harpoon that bacteria use to move around. And so this idea that structure turns into function, is heavily relied on in our own understanding, right? Like that's what we know from our day-to-day life. And that's usually how a lot of the proteins in our cells work, right? So it doesn't have to be these massive assemblies. It can also be smaller complexes like polymerases. It can be metabolic enzymes or other proteins. But all of these have this specific structure that essentially facilitates their function. So this is good, and the reason why this is good is because the cellular environment, the environment this proteins actually work in, doesn't look like this blank space. It's actually completely jam-packed with other molecules. And this environment is constantly changing. So over here, we see some images of cells doing their thing, right? Splitting, moving, blebbing, doing all kinds of things that cells do. And it's clear, even though we don't have a single molecule resolution in these images, it's very clear that the environment undergoes dynamic changes over short timescales. And so if we think about kind of the physical chemistry of what's happening inside the cell, we can think about changes to crowding, ionic strength, osmotic pressure, pH, viscosity, and even the composition of the cell, right? The concentration of the solutes that exist inside the cell. And so those folded proteins, the proteins that have this specific structure have a strong hydrophobic core held together by thousands of intramolecular bonds, have a surface area that's optimized for the surroundings, and so isn't really perturbed strongly. And so these folded proteins may not care. And this makes sense, right? Because decades of biochemistry done outside the cell taught us everything we know about these enzymes. And the buffer environments where these experiments were done is very, very distinct from the cell. But not all proteins are like this. And so intrinsically disordered proteins, which I abbreviate here as IDPs, but also IDRs, which stands for intrinsically disordered regions, are outliers in this respect. Even though they're outliers, they compose a large chunk of the proteome. So it's estimated that about 40% of the human proteome is composed of these intrinsically disordered regions. They play really critical roles in cell regulation, in homeostasis, in signaling, and of course in liquid-liquid phase separation. And so these proteins or these proteins regions exist in this rapidly interchanging ensemble, where the ensemble is kind of the collection of conformations that these sequences can take. And these ensembles, especially compared to well-folded proteins, have very few intramolecular bonds and have a high degree of surface area, as you can see from this trajectory here on the right-hand side. And so it's this confluence of few intramolecular bonds and high degree of surface area that really makes these sequences poised to sense and respond to their surroundings. And so this is really our entry point. This is really the question that we're interested in. It is, how does the cellular environment interact with these disordered ensembles? And so when we think about this, since I am at least trained as a chemist, we start thinking about the physical chemistry of this. And when you think about that, you realize that these disordered regions sense the environment through their surface area and then can respond to their environment by changes in their ensemble. So how does this take place? Well, first of all, we can think about the global dimensions of the ensemble. So these hairballs that you see here are essentially a few hundred conformations of disordered protein that are overlaid one on top of the other. And depending on the environment the protein is in, this can cause this ensemble to expand or to contract. And so when we think about the physical chemical model or mechanism behind this, you can think about the interaction of the solution with the protein backbone or the volume taken up by the protein. And this has been characterized for many years by people like Reckert and Timoshev and Bolin and Minton through the action of osmolytes, which act relatively uniformly on the peptide backbone by repelling the peptide backbone, through the action of denaturants, which act relatively uniformly on proteins by attracting to the peptide backbone, exposing it. That's why they denature it. Or through the action of crowding, where the protein tends to take up smaller volume when in a crowded environment. So that's one aspect, these global dimensions of the chain. And the other aspect relates to the local dimension of the chain. When I talk about local dimensions, I mean specific interactions within the chain that can be modulated by a change in solution. And so some classic examples of this would be electrostatic interactions that can be screened away by adding salt to the solution. This can be attractions or repulsions. It could be hydrophobic patches or aromatics that exist kind of within the sequence and can bring together different regions of the sequence depending on, what is the solution around them. And so, this is kind of the theoretical framework that we have to understand how ensembles might change. And a question we had kind of starting out is, how do different IDP sequences respond to similar changes in solution? And the idea we had is that we expect that there's going to be some kind of solution dependency, or sequence dependence here, because these kind of specific interactions vary with the identity of the protein. And so we started out by just looking at simulations. And so we used the Campari simulation suite with the absinthe force field that was developed by Rohit Papu. And together with Alex, with Alex Holhouse, we've developed a method by which we can essentially tune the interactions of the solution around the peptide. And so over here, what you can see is how the global dimensions change as we turn the interaction between the solution and the peptide backbone into something that's more repulsive. So the more repulsive the solution gets, the smaller our ensemble becomes, because the protein wants to kind of compact in on itself to reduce the surface area, right? Reduce the amount of interaction with this repulsive solution. And then when we started simulating more and more sequences here, so this is work done by graduate student Feng Yu in the lab. So what Feng did is he simulated about 100 different intrinsically disordered regions, and we normalized them so that all the radius of gyration start from the same point. And then we just turn the solution turn the solution more and more repulsive. And what's completely clear is that there's a wide range of behaviors that are sequence dependent, right? And so we have some kind of average behavior where the sequence undergoes some intermediate collapse as the solution becomes more repulsive. But then there's going to be other sequences that show almost no sensitivity, and then other sequences that are exceptionally sensitive. And so we found this really intriguing, right? This implies that there's some kind of sensitivity that's encoded into the sequence, at least as far as this global dimension goes. Of course, we also wanted to look at how these local interactions or local structure plays a part. And so for that, Feng has developed an algorithm to help us visualize the contacts within an intrinsically disordered chain. So over here, what we see is an intrinsically disordered protein sequence, and the red lines imply repulsive interactions. The green lines imply attractive interactions. We're not going to go into how we calculate this. If anyone's interested, I'm happy to talk more about this later. But then when we take this sequence, which over here is simulated in water, and put it into different solutions, we'll see that these patterns change. And so when we have a solution that's attractive to the peptide backbone, kind of like a urea solution, what we see is almost all of the attractive interactions go away. When we put this in a solution that's attractive to hydrophobic patches, that becomes even more pronounced. But if we put this in a solution that's attractive to polar patches, then you can see that a lot of these contacts actually persist. And so even though all of these solutions kind of have the same global interaction, you can see that very different structures may arise when the specific interactions are different. And so this doesn't just happen in this sequence. We can see that these long-range interactions are very prevalent. And here's just a collection of a few of the disordered proteins that we've simulated. Each one of these boxes has a different sequence. And you can see that the attractions and repulsions in each of these sequences are very different and also often span the entire range of the sequence itself. So this is all great, but like any computational person worth their salt, the question always exists: is this real? Right? Does this actually happen in the real world, or is this just an artifact that's a result of whatever idiosyncrasies exist in the force field that we're using? And so to answer this question, of course, we needed to take this concept and build an experimental system that will test it specifically. And so the way we chose to do this is using ensemble fret. And so before I describe the system, I'm going to pause here and just say that ensemble fret has been, for decades, gotten a really bad rep. And I'm really hoping to bring it a little bit back into vogue because, single molecule fret, time resolved fret, these are fantastic methods, but these are methods that are relatively expensive to implement. They can be extremely low throughput. And, what we lose in ensemble fret in quantitative accuracy, we gain manyfold in throughput. And we also have the added advantage in that we don't need really fancy fluorophores to do these ensembles. And so this allows us to use fluorescent proteins. And these are fluorescent proteins is just such a huge advantage, right? There's no extra labeling steps. It's amenable to just expression in the cell. I mean, we've really been capitalizing on the ability to use these. All right, so after that, the construct itself contains two fluorescent protein, a donor, which is M turquoise 2, and an acceptor, which is M neon green. And then between these, we ligate a disordered protein sequence. And the idea is that when we excite the donor here, we're going to measure a spectrum, and we do these measurements in well plates. So it's relatively high throughput. We can do this in 384 well plates and just get many of these experiments done quickly. And so what we get is generally two peaks. One belongs to the donor, the other to the acceptor. We can kind of get rid of artifacts by looking at the donor alone and the acceptor alone. And over here, the shaded areas show the fluorescence of each of these fluorescent proteins on their own. And then we can deconvolute the measured fret spectra using these two base spectra and get at the fret efficiency in a specific disordered protein sequence. And so if the solution is repulsive and contracts the sequence, for example, what we're going to see is that the acceptor fluorescence increases, and vice versa if the solution expands the sequence. So the acceptor emission is going to decrease. And so what we're going to be measuring is this E-fret or fret efficiency, which is essentially the fraction of photons emitted from the acceptor over all the photons emitted total. Okay, so this is great, but it's not without problems. And one of the main problems with any fret measurement really is that they're really sensitive to the length of the sequence that we're interested in, really sensitive to kind of this distance between the two proteins. And so our solution for this is to calibrate the fret based on a sequence that we can characterize very well. The sequence we picked was these glycine serine repeats. The reason why we chose these is that there's evidence, especially coming from Magnus Kjærgaard's lab, showing that these glycine serine repeat polymers behave kind of like theta state homopolymers, right? So these are polymers whose behavior has been well characterized experimentally previously, and we kind of understand how they work. And so when we go ahead and measure the fret efficiency of different lengths of glycine serine repeats, we see this really nice linear dependence. For the aficionados of polymer physics, this is not what's expected from just a plain homopolymer, but remember that this is not a plain homopolymer. Remember, we have our two fluorescent proteins on either side, and because of that, we get this linear behavior. And what this linear behavior lets us do is it lets us interpolate what the Fred efficiency of a GS linker would be for a sequence of any given length, assuming we have that length of link, right? And so we can now essentially predict what the Fred efficiency of a GS linker would be for any length here and use that as a comparison. And so when we do that and we replace our GS linker with another disordered protein, we can always say, is this linker now longer than a glycine serine repeat, or is it shorter than a glycine serine repeat? At least as far as the end-to-end distance measured by the Fret labels can tell us. And so that's going to be kind of our point of reference. So we go ahead and we start doing these experiments. And so over here, this is kind of the basic experiment that we started doing. So on the x-axis, we have the concentration of a solute. In this case, it's sarcosine, which is actually a pretty prevalent co-solute inside cells, and it's a precursor of several amino acids. And then on the y-axis, we have this normalized Fred efficiency. So this would be essentially the Fred efficiency divided by the Fred efficiency of a GS linker of the appropriate size minus 1. So when it's 0, it's exactly the same dimensions as a GS linker of the same size. When it's positive, it means the sequence is more expanded. Negative means it's more compact. And so over here, the Fred efficiency of the GS 32 GS repeats, of course, starts from 0 because it's exactly the same dimensions as the GS 32. But as we start adding sarcosine into this mixture, we see that the dimensions start changing. And so we're really encouraged by this. And of course, here is where we can really capitalize on the throughput of the system. And so we don't have to do this for just one sequence or one solute. We can do this for lots of sequences and lots of solutes. So we call these kind of graphs solution space scans. As you go across columns, you see that we change the identity of the solute that we're adding into the solution. And then as we go across rows, we see that in this case, we're just taking longer and longer glycine serine repeats. And so you can see that in solution space, right, different solutes are going to have very different properties. And some of these can kind of be predicted, right? So polyethylene glycols and phycol, which are generally thought of as polymeric crowders, tend to compact the sequence. Urea and guanidinium, which denature proteins, generally expand the sequence. And then we see some more interesting things with others. Especially notable is the fact that, for example, salts in these sequences have this non-monotonic effect. But overall, you can see that regardless of their length, the GS linkers behave in the same way. So the response to the solution space scan for glycine serine linker is the same regardless of length. And we're really happy to see this. It made sense, right? Because whatever local structures you can think of exist in the sequence, they're going to be kind of the same regardless of the length of the sequence. By the way, I forgot to mention the color coding in the background is simply proportional to the slope, right? So if the slope is really strong, then it's going to be a stronger color and vice versa. Okay, so this is great, but of course we want to move beyond these glycine serine repeats into real disordered proteins. And so the question is, does this happen in real IDPs? And so we took a few classic examples, and we picked these examples specifically because in each one of these, or at least in almost all of these, the structure of the ensemble has been shown to be linked to the function of the protein. And so when we took these 4 different sequences and we did the same solution space scan, we saw something very different. So of course, each of them still responds differently to different solutes. But now as we go across the rows, we see that the intensity of color is very different. And while some sequences are, for example, strongly compacted by PEG, others are barely affected at all. And so the response to the chemical environment in this case is sequence dependent. And so we immediately thought, maybe this could be this evidence of hidden structure that we saw in our simulation. So this is great, but we were still a little bit skeptical. And part of the reason why we're skeptical is the reason that I started out with, and that is that ensemble fret is problematic. So even if we get rid of all the artifacts in ensemble fret, all the bleed throughs and the cross excitation, and we manage to really calculate the fret efficiency accurately, which is difficult, there's also some model dependence. And this model dependence specifically comes when we want to move from fret efficiency into end-to-end distance, we need to use this R naught factor. And the R naught is generally treated as a constant. There's a lot of different factors that fold into it, but it actually depends on many parameters. And saying that it can just be held as a constant, especially in different solutions, is a strong statement. So another method that we can use to talk about the dimensions of a sequence is small angle X-ray scattering, or SAX. And the big advantage of SAX, even though it's much lower throughput than our FRET experiments, is that interpreting SAX measurements in terms of a characteristic radius, and specifically the radius of gyration of an ensemble, is really model free. So we can take a scattering curve. So over here on the y-axis, we have the logarithm of the scattering intensity as function of the reciprocal angle of scattering. And when we take this and we fit this to a line over a certain region, it's called the Guinier region, we get out as a slope the RG. So there's absolutely no model fitting used here. And so we're really excited about the possibility of treating these RG measurements as kind of the ground truth, right? As something we know that works, assuming our measurements are of sufficiently high quality. In the background here is a long-term, I guess, controversy dealing with the agreement between FRET measurements and SAX measurements in intrinsically disordered proteins. Again, there's a lot to talk about there. I'm not going to go into it. I'm just going to say that at least in our constructs, this has been resolved, and I'll show you exactly how that happens now. And so we start with independent measures of RE through FRET and RG through SAX in our glycine serine repeats. And what this is kind of what it looks like. Okay, so the blue symbols here are the end-to-end distance. The orange ones are the radius of gyration. And together with Alex Solhaus, what we did was we essentially said, can we make an all-atom simulation that's going to simultaneously match both of these observables? Because if we can, then that's at least one possibility of an ensemble that can exist for these measurements. And the answer is yes. So we managed to do it. So Alex, using an all-atom homopolymer simulation, has managed to tune the parameters in such a way that the simulation RG and RE match almost quantitatively with our experiments. And this was really exciting because the way these simulations worked, it turned out that kind of the behavior of these simulations is exactly how we expect glycine serine repeats to behave. So this was really encouraging. And the next question was, can we match this also in actual IDPs, right? These are just homopolymers. So what happens if we now take a heteropolymer, change the sequence? Can we still get this interesting relationship? And so what we do next is just plot the end-to-end distance versus the radius of gyration. And you can see that the glycine serine repeats just fall on this nice diagonal here. And what we do next is we measure a couple of heteropolymers. So we measure P53 NTAD and the BHD domain of Puma, which we've seen before. And when we put those on the graph, well, not really. So not only are they not on the diagonal, which, okay, I guess that can be expected, they also have almost the same RG, but very, very different end-to-end distances. And one is below the diagonal line of the homopolymers and one is above. And so this is not really what we expected to see, right? This implies that something here is very, very different from the glycine serine repeats. And so again, our go-to are these long-range hidden structures that alter the ratio that exists in these heteropolymers, but not in a homopolymer, and alter the ratio between RE and RG. And so we thought, how can we test this hypothesis? Well, one way to do that would be to take the sequence of one of these IDPs and scramble it up. So we took the Puma BH3 domain, you see the sequence over here. We scrambled it up. Our visualization implied that the long range interactions kind of differ dramatically between the different sequences. And what we did next is we repeat this correlated Fred and Sachs experiments. What we see is that we see the wild type here, and then we see the 3 scrambles here. And even though the amino acid composition is exactly the same, the length is exactly the same, we get very, very different behaviors of RE and RG. So not only that, but the scrambles also show very different solution sensitivities. So if we do our solution space scan, which is now a little bit more limited, but once again, we see that, for example, the wild type has a very high sensitivity to crowding and denaturance, whereas some of the scrambles are essentially unaffected by anything. All right, so to conclude, I think, or I hope that I've managed to convince you that by combining Fred and Sachs, we're starting to reveal these hidden structures that exist in intrinsically disordered protein ensembles. I've shown you that these long-range interactions can be prevalent in these ensembles and often, largely determine their shapes. I've shown you that these long-range interactions can respond to changes in the solution, and also that the changes in the solution can in turn inform on what hidden structures are there. And so the bottom line is that at least in vitro, the physical chemical environment matters, at least as far as the conformational ensemble of the IDP is concerned. And so what we're doing now is we're adapting this methodology for functional studies. So we're looking at how these changes in ensemble translate into changes in protein binding and phase separation and post-translational modifications and so on. So this is all great, but here's the place where we remind ourselves that this all happened in vitro, right? And I started out my discussion about how the cellular environment changes and it matters. And so really the question is, does this kind of effect happen inside the cell? And so here again, we capitalize on the fact that we're using fluorescent proteins as Fret labels. And so we can simply, transiently transfect these plasmids into mammalian cells. And this is what we do. So generally we use U2OS cells, though we've now used a few cell lines. The results are to a large extent similar, even though there are notable differences sometimes between cell lines. And what we do is we look at the emission of the donor and the emission of the acceptor through filters on our microscope. And this, of course, also allows direct comparison with in vitro experiments because, while we don't have the full spectrum in our microscope, we can just take specific windows in our spectra and then calculate the donor and acceptor fluorescence in the same way we do in our microscope. Okay, so now let's start looking at it, right? And I should point out that these experiments are actually much, much easier to perform than the in vitro experiments because there's no purification or expression purification process necessary here. So the first thing I want to show you is that glycine serine repeats inside the cell behave kind of like they do in vitro. So we're now going to transition to a y-axis that shows the donor to acceptor ratio. So as this increases, that means the donor is further away from the acceptor. And you can see a linear scaling of D over A as we increase the number of residues in our glycine serine repeats. This is what the same thing looks like in vitro. You notice there's a change in scales. This is expected simply because the sensitivities of the different imaging devices varies. We can also start looking at IDPs. So for example, we repeated our Puma and Puma scrambles experiment inside the cell. So again, you look at D over A here, you see the wild type S1, S2, S3, and here's the comparison to the same thing in vitro. And you can see that the trends persist, right? So wild type is the most compact here, S1 is the most expanded, and so on. And this is, by the way, the average of, over 10,000 cells in each of these. So, these hidden structures that exist or that we think we're measuring in vitro also persist inside the cell. Of course, we're not limited just to this. We can now look at a whole range of different proteins. And so we tried different naturally occurring IDPs inside the cell. You're going to see them like this. So over here, this is kind of a violin plot. The little points and the little circles in the middle are individual cells. So we're taking whole cell averages. The square is the average, and you get the standard deviation here and the distribution denoted by the violin. And then again, the D over A, the fret signal is normalized to the GS linker of the equivalent length, so that when it's 0, it's going to be more, it's the same as a GS linker. When it's negative, it's more compact. When it's positive, it's more expanded. And so this is the picture we see for a few different disordered proteins that we tried out. Over here on the left-hand panel is just the length of that protein, right? So it's arranged by length, the shortest one at the bottom, the longest one at the top. And you can see that it's very much length independent. So some of these sequences just in cells that are in normal conditions behave as the GS homopolymer equivalent would, but others show more compaction. We actually have not yet observed the case with more expansion, and that's interesting. And again, we have thoughts on why that happens. But what it means is that, right, these different sequences are going to have, in certain cases, some hidden structure that would cause them to compact more than a homopolymer otherwise would. Okay, so this is a good start, but what we really want to do is kind of do our solution space scan, right? We want to kind of kick the cellular environment around a little bit and see what these sequences do in response. And so, how can we actually do that, right? The cellular environment is very complex, like we already said, but it's also adaptive, right? So when we do changes to the cellular environment, the cell's going to kind of try to counteract them. And, we're really interested in, again, the protein behavior. So kind of devoid of the context of, the regulation machinery or the homeostasis machinery of the cell. And so how can we decouple the physical response of the cell, the physical response of the proteins from the biological response of the cell itself? And so the best way to do this is the realization that the time scales are drastically different, right? The physical chemical adaptation of a protein, right? The change in ensemble structure happens in milliseconds or less, right? Especially in disordered proteins. The conformations are sampled extremely rapidly. Whereas biological adaptation, right? For example, phosphorylating the protein or, causing some cascade that would counteract the stress or the perturbation that we're doing to the cell, that takes something on the order of minutes, like 20 minutes or more, especially if we're thinking about transcriptional response, right? And so what we do is we try to resolve our experiments very quickly. So something like 2 minutes or less. Generally, we actually do this in less than 2 minutes. We actually do this in less than 1 minute. And our perturbations are very rapid. And the perturbation I'm going to talk about today is osmotic pressure perturbation, which is historically something that I've been really interested in and is actually gaining a lot of attention recently as a fantastic way to probe kind of the biophysics of the cell. And you can see here that these osmotic changes happen very reproducibly and very quickly. And so it's just a really good handle to perturb the intracellular environment. So what we do is we measure the D over A, right, the fret signal inside the cell before and after osmotic perturbation, and we plot it like this. So over here on the y-axis is the fold change in D over A. So the fold change would be after perturbation divided by before perturbation. And on the x-axis, we see the osmotic pressure after the perturbation. So on the left-hand side, we have hypoosmotic pressures. So the cell actually expands under those conditions. And to the right of 0.3, we have hyperosmotic conditions. So the cell actually compacts. And isoosmotic conditions are 0.3 osmolars. And once again, we have this calibration to a kind of the basal level. And so if it's above the 0 line, the osmotic challenge will expand the sequence. And if it's below, the osmotic challenge will compact the sequence. And so we start out with just our glycine serine repeats. And so over here, we have increasing lengths of glycine serine repeats. You can see in all of them, when the cell volume expands, right, the environment becomes more aqueous, and overall, it looks like the polymers expand. And then when the environment turns hyperosmotic, the cell compacts, water is pushed out, everything becomes more crowded, the glycine serine repeats contract. And so this is kind of what we expect. We were very happy to see this. This is also the trend we've seen in vitro. I don't show that here, but it was kind of apparent in the solution space scanning slide. And we can even get some insight showing that, for very short glycine serine repeats, the slope exists but doesn't really change. And the change really happens as sequences grow longer. But then this picture gets really interesting when we start looking at heteropolymers. And so we do that, we see trends that not only are extremely sequence dependent, but also simply cannot be explained by any current I guess, biophysical theory that we usually use to explain in vitro observables, right? So you can see sequences that behave, somewhat similar to the GS linkers like NUP15 and things are in the way, and CMYC. But then we have stuff that show, nonlinear response like P27. We have stuff that's extremely insensitive like NUP49. And maybe most interesting, sometimes we get effects that simply contradict what we expect from crowding, right? So we have sequences that compact under hypoosmotic conditions, but expand under hyperosmotic conditions. And this is absolutely fascinating to us. And so, since crowding models and even, osmolite or denaturate models can't really explain this, what we really need to look at now is the biology, right? And that's kind of what we're doing now. So we're looking at post-translational modifications. We're looking at protein-protein interactions that might actually affect the dimensions of these ensembles. And so to conclude this part, disordered proteins maintain their hidden structure inside the cellular environment. These ensembles do respond to physical chemical changes inside the cellular environment, right, to changes in the cellular solution. The sensitivity of these IDPs can sometimes depend on sequences, but sometimes depends on more than sequences. And so this is, like I said, kind of the stuff that we're starting to look at now. So with that, I'd like to talk a little bit about the implications of all of this. And so the point we started out with is that the intracellular physical chemistry changes routinely. And this is exemplified beautifully when you look at any, any imaging of mitosis, right? The changes that happen in the cell, the nuclear envelope breakdown, the change in shape, the change in the cytoskeleton networks, all of these are dramatic changes to the physical chemistry of the cell. And they happen every single day, hundreds of millions of times. And so we hypothesized that certain IDPs are really evolutionarily designed, not just poised, to sense and respond to these kinds of changes. And when you think about it, this is such an elegant, rapid, and efficient way to sense and respond to changes in the cellular environment. And so zooming in now and thinking about phase separation, right, condensate-forming IDPs may have long-range hidden structures, right? These structures can also exist in condensate-forming IDPs and even in LCDs. And we actually show this for a couple of them, Fib1 and A1 LCD, where it's clear that A1 LCD, despite being actually longer than Fib1, shows a more compact conformation here. Not only that, these condensate-forming IDPs can have a wide variety of sensitivities to their surrounding environments. And these two sequences happen to show this beautifully, where A1 LCD is extremely, so this is an outlying insensitivity to changes in cell volume, whereas Fib1 has an outlying sensitivity to this. Not only that, Fib1 acts completely opposite of what we expect from crowding, right? As the crowding decreases, the sequence compacts. As the crowding increases, the sequence expands. So this is really fascinating stuff, and we're trying to correlate this now with the CSAT and other parameters in phase separation of these two proteins. But what's important to remember is that many condensates, are formed in response to some perturbation to the cellular environment. And over here are two collaborations, one with Aaron Gittler and Sue Rhee, regarding FLO1, which is a plant protein that actually phase separates or condenses when the environment becomes hydrated. The other one is YAP, so this is a transcriptional coactivator that phase separates again in response to osmotic changes around the cell in collaboration with Jennifer Lippincott-Schwartz and Dan Fan Kai. And so, the fact that condensates are so responsive to these physical chemical changes in the cellular environment raises a lot of open questions, right? How does the cellular environment actually regulate this liquid-liquid phase separation? How do the hidden structures inside these proteins determine the condensate properties, right? Whether it's the tendency to form condensates, the material properties of the condensates, or the specificity of these condensates. Another question is, how do these ensembles change, right? How do these hidden structures change between the dense and the light phase? And how all of this is affected when the cellular environment becomes misregulated. And so that leads me to the next issue, which is that this finding can have really important implications when it comes to pathology. So pathology is a major driver of changes to the physical chemical environment of the cell. And the examples are endless. Probably the most famous one is the Warburg effect in cancer cells. I mean, that's been characterized, over a century ago, and shown to change a whole bunch of different parameters inside the cell, from the pH to the concentration of metabolites. But this kind of effect happens in viral infection, in neurodegenerative diseases, and many other different cases. And so if IDP sequences are meant to sense and respond to routine changes in the cell, we got to ask what happens to their function when the environment is strongly perturbed. And kind of a direction for us looking ahead would be to look into this as a mechanism by which diseases like cancer can progress even in the absence of mutations, right? Just by a misregulated intracellular environment. All right, so we're almost done here. Kind of a final summary. Long-range hidden structures in IDPs are prevalent. The hidden structure can be tuned by changes to the intracellular solution. That's why we're thinking that the solution is the solution, right? That's our initial pitch. And function, this is already known, that function can be regulated through local and global ensemble changes, and that IDP function may be impaired in a pathological cellular environment. All right, so now come the thank yous. So see the animations here are a little out of sync. I, first of all, really want to thank the graduate students who did all the work here. So like Mark said, the lab is relatively new. Most of the graduate students have been here maybe 2.5 years. And the work that we've accomplished since then is really phenomenal. And so Karina Guadalupe, a third-year graduate student, has done almost single-handedly almost all of the live cell imaging data and analysis that I've shown you. Feng Yu has done all the computational work, including the visualization of long-range interactions. And David Moses has developed and taken all the in vitro data. And then these are other lab members who all contribute either intellectually or in actual help to the work, and our collaborators, Alex, who's done a lot of the simulation work that I've shown you, and Eric Martin, who's helped extensively with some of the SAX measurement. Again, a lot of this is done through kind of infrastructure that you can apply to. So we've done SAX measurements on SLAC, Argon, and ALS, computational work on Exceed and the Merced cluster, and funding from NIH and CCBM. And before I finish, I'm just going to say that the lab is looking for a postdoc. So Sorry to put this shameless plug at the end, but
Okay.
if you're interested, yeah, there's a lot of freedom, diverse and supportive environment, 4 years of funding, super close to Yosemite. And by the way, here's a recent lab trip where I almost killed my lab. That's a story for another time, to Yosemite. And yeah, with that, I'd like to thank you for listening and take any questions.
Now, thanks very much. Wonderful talk. And by the way, if you could get, if you could scarf up some tickets to Half Dome, you know,
Ooh.
I'd even sign up for the postdoc. It's so hard to get there. So hard to get the Half Dome tickets, you know?
Yeah, over here, a lot of the faculty have regular reminders on the day that the signups start. That's the only way to do it.
Exactly. No, fantastic talk. Thank you. And lots of interesting questions coming in. I know we're close to the top of the hour, so I'm not sure, you know, who's able to hang around and who's not. But maybe if Charlotte is still on, maybe your question I thought was a good one. If we could start with you.
Yeah, hi, can you hear me?
Yeah, hi, Charlotte.
Hi, great talk. So I guess I had a couple of questions, so I'll try and go through them a little bit quickly. But the first question is, have you looked at if there's any relationship between how cells or how proteins respond in cells and their cellular function, like if proteins in one compartment or one sort of function have a consistent activity?
Right. So, I mean, you're talking about some kind of like Go anthology or something like that and see if it breaks down. Yeah, I mean, that's a fantastic suggestion and one that we're looking into. I think that right now, the data set that we have is probably not extensive enough to really get good inferences there, right? I mean, usually you get strong correlations when you have, you know, large like genome scale or proteome scale data. Over here, you know, we're ramping it up, but we're still on the like 10 to 15. So I don't think that currently there's any strong correlations there, if that makes sense.
And then my other questions were about sort of the more the in vitro experiments, because you talk about compaction a lot, but I was wondering if you had any sort of insights into what the nature of that compaction is. Like, are you forming similar types of structures, like sort of within the intrinsically disordered domain? And then sort of relatedly, can you sort of induce and reverse compaction, and are those cycles similar in any way?
Right. So I'll answer the first part. So the compaction, from what we see at this point, can be driven by different motifs, right? There could be different combinations that would bring about this compaction. And, you know, our way to know what it is, I think, really hinges on our interpretation of the solution space scans, right? Because say the compaction is kind of abolished by adding salt into the solution, right? That would mean that it's driven by electrostatics. Whereas if the compaction happens with, or sorry, the compaction is abolished by even a small amount of say urea or guanidinium, it may imply that these are kind of more dispersed along the chain and not as strong as electrostatic interactions would be. So this is really a direction that we're looking into, kind of zooming into the specific sequences and maybe with the use of mutations even trying to predict what the driving forces for compaction or expansion might be. And so in terms of the cycles, do you mean like cycles, like you talk about live cell, or maybe help me understand exactly?
Yeah, I guess it might make more sense to do experiments in a live cell, but even sort of in a fret type situation where you like maybe replace the buffer with one that promotes compaction and then reverses. And if you see sort of like maybe it becomes less dynamic when you switch buffers back
Right,
and forth for
right,
some
right.
versus others.
So kind of like hysteresis as a result of like what we see when we do thermal denaturations, right? Yeah, that's
Right,
actually a
yeah.
fantastic idea. We have not done this, right? The way we do this is on a plate reader, so we just do one and done. But that's great, and we should probably look at that. Thanks.
A couple of good questions also from Amira. You know, the first one, I don't know if it's still on the line, but I love especially the first one, the age-old challenge of distinguishing, you know, results in cells from in vitro, you know, in particular in the context of the work that you described today. You can probably see it in the chat.
Okay, let me see, because I haven't looked at the chat at all.
About the in vitro
I've
versus.
unmuted her. She's ready to talk.
Oh, great.
Oh, perfect.
Hi, great
Hello.
talk. Hi. The first question, it was regarding the perturbation that you have done in vitro and in cells, because in cells you have interactions of these proteins with other molecules, DNA or other proteins. So do you have an example that behaves completely different in vitro after perturbation than in cells?
Yeah, so essentially every single time we've seen a protein expand when the cell volume decreases, that's a result that cannot be reproduced in vitro, at least as far as we expect, you know, a PEG solution to be a more crowded solution, right? Which is, there's problems with that assumption. But that like I said, there's absolutely no, you know, there are solutions that would expand the protein, but not in the context of a compacting cell. And so, yeah, I mean, what you said is absolutely right. That's the directions we're looking at. So we're starting to run these in situ cross-linking experiments where we cross-link, you know, the entire contents of the entire proteome of the cell and then pull down on our specific proteins and see what gets attached. And so then we can do that in different osmotic perturbations and kind of see how that gets modulated. And maybe in the context of that, I mean, we had a 2017 PNAS paper where we show that these changes in volume can actually cause protein-protein complexes to form more readily. And so that very well might be something that's happening.
Do I have time for another question? Or?
Yeah, your other question's also one of those classic questions, you know, cytosol versus nucleus. It's a great question. Why don't you go ahead?
And so I was wondering with the GS repeat proof in cells, if you tag it to the nuclei or cytoplasm, if you see a difference with the osmotic pressure changes.
So actually, Diana, one of the undergrads in the lab, is doing that analysis like now. So
nice.
we'll have that soon.
Nice, thanks.
Beautiful, beautiful. How about Kibyam Hong, if you're still on? Maybe not. But the question in the chat is interesting. You know, the FP fused IDPs, might they exist as multimeric forms?
Yeah.
And
so
the measurements you're seeing.
Right. So that's a good question and definitely something that we were concerned about. Let me just find this slide. Yeah. So over here, this is the untethered fluorescent proteins. And you can see that. So what we do is we use them in the same concentration that they would be as a tethered construct. And you see that when we break that link, the Fred efficiency is 0. I don't have this here, but we also do some concentration dependence of the Fred efficiency, and there is none. Again, as we go to higher and higher concentrations, this might happen. Inside the cell, again, we do similar controls, and we do get some artifacts in cells that have extremely high expression levels. Again, remember, this is transient expression, so expression levels are varied. We generally filter out, you know, over a window of expression. So we want kind of intermediate levels of expression. So cells that are expressing a lot, we generally filter those out. And that's kind of our way to circumvent this, right? Our main control is, inside the cell at least, is looking at correlations between the Fred signal and the direct acceptor signal, which is only proportional to concentration. And our basal hypothesis is that, that's kind of like concentration series when you think about it. Our basal assumption is that we're measuring the monomer, and so there should be none, right? So we kind of, that's kind of the control we have on that.
That's great. So Emery has an interesting question about how the properties of the IDPs affect your measurements. Why don't you go ahead, Emery?
Hi, awesome talk. So I was just curious if you've done any looking into how the sequence space of the IDPs you selected in this study, how those may scale or trend with the measurements of RG in the different conditions. So like if you're looking at net charge per residue or hydropathy, how does that, if at all, does that impact what the RG looks like?
Right, thanks. That's a great question. So, basically, the best way to answer this is to think about the scrambles, right? The global sequence properties of the scrambles are all the same, right? Net charge per residue, the average hydropathy, that's all going to be the same. And I've already shown you that, those give very different, again, I'm trying to look for that slide. Yeah, so those give very different, both RG and RE results. Then if we try to correlate this with kind of patterning behavior, there's usually that is more successful. I don't have this because it's very preliminary yet still, but for example, this was kind of surprising, but the behavior in the cell seems to correlate a lot with actually hydrophilic patches, right? So the hydrophilic patches showed the strongest correlation with the basal dimensions of the sequences inside the cell. So this is definitely something we're thinking about in a fantastic question that I still don't have a straight answer for,
Oh, cool. Thank you so much.
Mm-hmm.
Great. And how about Yifan?
Yeah, thanks for the talk. So my question is just a fused fluorescent protein that's supposed to increase the solubility for your IDP fusions. Therefore, how that can impact the IDP phase transition behavior.
Right, that's a great question. So the way we're approaching this right now, and you can think of this as a disadvantage, but I actually think of this as kind of an advantage. The reason is that, and here, let's bring up this slide here. The reason is that adding those FPs kind of ensures that we're looking at the monomer, right? And so if our question is, what is the behavior, or how does the behavior of the monomer then get translated into CSAT or some, collective variable, then we can simply look at the behavior of the monomer and then do some correlative experiments with the unlabeled sequence to see if it makes sense. Does that answer the question?
Yeah, kind of. I have a follow-up question. So when you do this kind of frame measurement, you are investigating the intro-protein interaction. So how this intro-protein interaction can be correlated to this kind of phase separation behavior specifically? Are they like competitive mechanism?
Right. So that's a great question, I think. So first of all, the dimensions of a sequence can correlate with CSAT, right? I think Eric Martin and Alex Holhouse showed this in their science paper last year. But in this case, especially when you think about these long-range interactions, right? If we think about the sticker and spacer model, then it's definitely possible that under certain conditions, stickers will stick to stickers intramolecularly and kind of compete out with intermolecular interactions. And that's one of the directions we're thinking about.
Yeah.
I think maybe we're at a good stopping point. So we're about 10 minutes over, and we are starting to lose folks. So I think maybe this is a good spot. What often happens, Shahar, is that people will have questions later. So don't be surprised if you get pinged by different people saying, "Hey, I loved your talk, but I was thinking some more about it," and so that happens quite often.
All right. Yeah, looking forward to it.
Bede just said he's going to email you 50.
I saw that.
So.
And we'll have you email some of the answers to some of the other questions that you didn't have time to answer too. So we'll
Mhm.
get
All
back
right.
to you soon about the chat.
Fantastic.
Beautiful. Well, thanks again. Thanks, Shahar, and thanks
Thanks
everyone.
so much for having me. This was absolutely great.
Really stimulating. I'm thinking lots of good stuff here. All right. Thank you, everybody.
Thanks, Shahar.
Okay. Take care. Okay.
Bye-bye.
Bye-bye.